Good morning, my name is Rachelle Coicou, and I'm here to introduce to you Dr. Plinkett's lab and to speak of my summer research project entitled Characterization of Primary Cultures from Adult Zebrafish Brainstem and Their Interactions with an Inhibitory Chondroitin Sulfate Proteoglycan-Rich Environment. It is well established that fish and amphibians are capable of regenerating severe descending axonal nerve tract and the central nervous system. This means that when mammals injure their spinal cords, as you can see here, they're incapable of passing over the CSPG site, which is the injury site, so they're paralyzed for life. In contrast, mammals are able to pass over that site and in less than 2 weeks recover 80% of their swimming abilities. What is a chondroitin sulfate proteoglycan-rich environment? CSPGs are a large family of glycoproteins. The core domain is made of proteins with repeating chains of disaccharides. In mammals, CSPG play a key role in the spinal cord injury because the area around the injury site is saturated with these molecules. They're believed to be one of the main factors causing inhibition of axonal growth. The main purpose of Dr. Plinkett's lab is to study the molecular biology of neuronal regeneration in zebrafish after spinal cord injury and to explore the behavioral response of zebrafish neurons to inhibitory CSPGs. Our main method of research is culturing. We culture the lower part of the brain, which is the brainstem, that contains the nerves which connect the brain and the spinal cord. Dr. Becker's lab in the United Kingdom was able to prove that the brainstem contains a nuclei with cells of both high and low regenerative capacity. We hypothesized that our brainstem cultures will have different neuronal populations and that each will respond uniquely to each CSPGs. And we believe that the ones of low regenerative capacity will be the ones repelled by the CSPG barrier, but the ones with high regenerative capacity are those cells that are able to grow past that barrier. The way we culture the brainstem is by dissecting the brain and taking out the brainstem. We culture them for 3 to 7 days in complete media, and after 48 days, we replace the complete media by 2 ways. We either replace it by complete media or by serum-free media, which is a synthetic media. After 3 to 7 days of culturing, we use immunocytochemistry to stain the brainstem, using DAPI staining for the nucleus and tubulin staining for the cell processes and the body. After staining the cells, we use a Zeiss fluorescent microscope to see the cells, and we trace them using an ImageJ software. As you can see here, as we expected, the cells grow with time. This is a 1-day cell. This is a 3-day cell, which has its axon in one dendrite. This is a 7-day cell, and this is a 14-day cell. Each experiment is conditioned, has 3 experiments and about 100 cells that we later trace using the ImageJ, which allows us to quantify our data. This chart explains how the cells grew, and here we have error bars. The error bars are obtained by using a statistical calculation called the standard error of the mean. This allows us to chart it, and we can measure how the cells are supposed to grow in case the experiments need to be repeated. This is different responses to CSPGs that we obtained in our cultures. We saw that some cells, as we hypothesized, are being grown inside the CSPG barrier. Some cells are able to cross in, which we believe are those of high regenerative capacity, and some cells are being repelled by that barrier. The way we measure these cells is by measuring 500 micrometers inside and outside as an area. We count the cells one by one, and we score them. The ones crossing out, the ones growing inside the barrier, the ones that are able to grow inside from the injury site, which are the main ones that we're studying, and the ones that are being repelled. So after scoring them one by one, we're able to come up with this data sheet and the percentages. We realized that in complete media, the percent repelled was 52%. The 24% were able to cross into CSPG barriers, 8% crossed out of the CSPG barriers, and 16 uniquely extended within the CSPG barriers. Similarly, in serum-free, 46% were repelled, 28% crossed into the CSPG barrier, 9% crossed out, and 17% extended within the CSPG-rich area. What this means is that there's no statistical difference between the undefined complete media, which has components that we're not sure of completely, and the synthetic one, which is basically scientific, which scientists made. A manuscript is currently being prepared for the publication of the establishment of brainstem culture, and you will hear more about Dr. Plunkett's lab by the further talks of these students. I would like to thank Dr. Plunkett and Dr. Alexis Castillo for allowing me to work in their labs, and my mentors, Fran and Ma'am, and also the MSCIP and the U.S. Department of Defense for funding our research.
Any questions?
So what was the difference between your complete media and your serum-free media that you mentioned there are unknown components?
The complete media is made of bovine serum. The bovine serum is basically cow blood. So we're not completely sure of the components that are allowing the cells to grow. The serum-free media is scientific made. The scientists prepared it in the lab. But what we realized is that in the serum-free media, by itself, the cells didn't grow. They eventually died. So what we do is for 48 days, we keep them in complete media, and then we transfer them on the 3rd day in serum-free media.
Could you explain what's the statistical method that you use to analyze your data?
Well, I know that, I'm not too sure.
So you mentioned that you're using some particular package, and you mentioned like a keyword that you use to analyze your data. Do you have any idea what that thing is doing to your data? How far the package that you use can manipulate or bias the result of your research?
I know that we had two options. We either use the standard deviation of the mean, or we use the standard error of the mean, or we use standard deviation. The difference is that standard error of the mean allows us a smaller range to chart. And that's why basically we picked this.
And what do you mean by the standard error of the mean and the standard deviation?
I know they're both statistical calculations, and we use something online, a software online, and we plug the numbers in and we find them. Any more questions? Yes.
Yes, please. Would you be able to go over the very first slides? I'm sorry, I was not able to follow the very first principles you were trying to establish between mammalian and another system.
You want me to start over?
Yes, if you don't mind going over those.
The CSPG, there's a CSPG barrier found in the injury sites, which basically grows after the injury. What we realize in mammals, the cells are not able to grow past that injury site. The whole area around the injury site is saturated with these molecules, and the cells are not able to grow past it. And in the zebrafish, they cross over. That's why we're basically paralyzed after spinal cord injury. And the zebrafish, they're able to not only cross over it, but it only takes about 2 weeks for them to get about 80% of their swimming abilities back. What we believe is that in the zebrafish, those CSPG molecules during regeneration are either absent or reduced, and that's why they're able to cross over that barrier. So we're trying to find the gene that's responsible for that crossing over. Yes.
I think you did a great job. Did you compare the same area of the brainstem in all the cases when you were fluorescently labeling the,
Yes.
the neurons? Do you know what area of the brainstem that was?
That was a
Okay.
Any more questions? Thank you.